Our brains are extraordinary. They take in waves of light and translate them into our rich experience of the world around us. This magical journey begins with our eye's superheroes, the retina. Now, light doesn't just waltz straight to the photoreceptors. No, it has to go through a sea of cells in the retina before it hits the dance floor. That is, the photoreceptors at the back of the retina. Once our little photons reach the photoreceptors, the magic happens. They spark activity that tickles the bipolar cells. The bipolar cells, like any good middle manager, pass the message along to the ganglion cells, the ones at the front of the retina. These cells get together and form, yes, you guessed it, the optic nerve. This is the information superhighway to the brain. But wait, we're missing some key players here. Enter the horizontal and amacrine cells, those social butterflies of the retina, managing the lateral interactions. Now picture each bipolar and ganglion cells as the conductor of its own tiny orchestra responding to a small patch of photoreceptors, the receptive field. Each cell orchestrates a beautiful symphony in response to the light. These receptive fields are like little targets with on-center and off-surround, or vice versa. These are always antagonistic, like rival siblings, constantly trying to outshine each other. This rivalry is the result of lateral inhibition, where excited neurons reduce the activity of their neighbors. This creates a contrast in stimulation that enhances the brain's response to discontinuities, or basically any changes in the environment. This mechanism is one of the most fundamentally important in the brain for perception, for every process. Let's imagine a simple scenario where light shines on a photoreceptor. It sends a signal to the bipolar cell, but at the same time, it also sends a signal to the horizontal cell. The horizontal cell, in turn, inhibits the activity of the neighboring photoreceptors. This means that the photoreceptors directly in the light signal more strongly than their neighbors. This difference in signaling enhances the contrast at the edges of light and dark areas, making it easier for us to perceive boundaries and edges. This is lateral inhibition in action. Okay, let's see what happens with an on-center cell when light enters the ring of its receptive field. Get ready, it's showtime. In the spotlight, our on-center cell sees an increase in activity, but introduce light to the edges and suddenly it's not so keen to perform. Now imagine we flip the script and shine a light on an off-center cell. The response is the opposite. The spotlight inhibits activity, while light at the edges gets the cell buzzing. Ganglion cells are the pickiest of performers. They prefer a well-placed spotlight or a boundary line to a full-stage illumination. It's all about that contrast, or as perceptual psychologists call it, illuminates contrast. Let's take this ganglion cell for a walk across a light-dark boundary and see how it responds. You see, as we move the receptive field, the cell's response rate changes. This ability to detect contrasts helps our brain construct information about edges and shapes. It's like fitting pieces into a puzzle, and voila, we perceive the world around us. So why does all this matter? Our retinas are essentially emphasizing the crucial bits of the visual scene, the boundaries, the edges. I can't overestimate how early and how much filtering and processing happens right at the start of vision, for all the senses actually. The cells in the retina give the brain the highlights of the story and let our brains fill in the details. In our next video, we move one step closer to talking about how the brain converts these signals from the retina into your visual perception of the world. I hope you had fun on this retina roller coaster ride. I'll see you in the next video.